All right, I got the very clear signal from backstage, where it's not really the backstage, they're just here, front stage, middle stage, that we can start. So can I please have quiet? Oh, yes, you do follow instructions. This is wonderful. I can also see some of you strategically decided to stay by the buffet, which is great. So you can listen to us from the table. But I would please like to ask you to be in conference mode now, because we're back to the talks. We're back to presenting the research streams and the fantastic, fantastic talent that we've got gathered here today. Just to wake you up after lunch, I'm going to ask you a question. How many of you have seen Guillermo del Toro's Frankenstein yet? I'm not sure it's in cinemas, actually. How many of you have read Frankenstein? Yes. How many of you see the parallels to Frankenstein that was written 208 years ago now to today? Ah, fantastic. You see, I'm an archaeologist, so I enjoy making these kind of trivia connections. But when Mary Shelley wrote Frankenstein at the age of 19, which is nuts, laying the basis for basically the sci-fi genre as we know it, it was an amazing time in academia. You had galvanism that was rising up. People were fascinated by electricity. It gave rise to these fantastic phenomena like the occult. And people started really wondering, what if you harness electricity and combine it with humanity if you're going to get artificial life? And that is exactly what Frankenstein is about. Now, Frankenstein goes down a slightly dystopic path. Most people in it die, and it's all a bit sad and tragic, typical Gothic horror. But actually, this desire of humanity to kind of harness the modern technologies for life itself and to understand why we behave the way that we do hasn't really left us in 200 years, 2,000 years really. Some of the earliest ancient Greek myths are about automatons. So I think that today what we're really, really looking at is breaking down the silos of academia, looking at interdisciplinary science and how we can harness the power of machine learning and artificial intelligence that's staring at you from every page in the newspapers with more soft ideas about understanding how the brain works, how we transfer knowledge, how humans function. And I thought that I was out of place as an archaeologist in this place, but when I was talking to Ara just before the starting of this panel, he mentioned the Rosetta Stone, and I thought, yes, these are my people. So I'm very, very excited to be here. We heard some fantastic, fantastic presentations until now, and we're going to dive straight back into them. I would like to invite the first speaker of this session, Saltuk Eyrilmez from ICRC RECETOX at the Maastricht University, to talk to us about the recent progress in drug screening for APOE or APOE. It's a lot of abbreviations, I'm sorry, I got most of that wrong, but please, round of applause. And also, use Slido, don't be shy, ask him questions. Thank you.
Thank you very much, very nice introduction, and I'm really happy to be here talking about our progress, our scientific progress in front of this very wide variety of people from different fields and different countries. So I'm Saltuk Eyrilmez, I'm coming from Maastricht University and I'm working in ICRC as a postdoc researcher and mostly working on APOE protein, which you have heard many times during the presentations. So first of all, I will start talking about Alzheimer's disease and role of APOE isoforms in Alzheimer's disease, and a little bit dive into more structural point of view of these protein-ligand interactions with more like a mode of Lego or Tetris, but in nanometer scales. So then I will try explaining how are we doing our search for drugs for these targets and results in summary. So first of all, you have already listened many aspects of Alzheimer's disease. So there are too many different reasons for Alzheimer's disease to develop, but the main problem is that this neural decay and loss of autonomy. So this is really an important thing which affects the quality of life of the people. And it's really projected for 2.5 folds for 2050. So this is a very serious one. As from the discussions, you also saw that it's a multifactorial disease which has primarily by age, trauma and genetic reasons. So firstly, we have, in our research, we have mostly focused on APOE polymorphism. So just for checking what APOE is, kind of a protein like that, made of different domains. One is the receptor binding region, and another one is the lipid binding region. So this works as in the lipid metabolism and transfer in the brain. And there are lots of mutations on that protein. Over 30, counting with the minors, mutations in the protein, but one of the isoform, APOE, is the APOE4 is the most problematic one, which we have a mutation on the receptor binding region of the protein. Just to check what it does actually is the lipid binding part binds to the lipids, and then after that this receptor binding region acts in a way forming some sort of structure with the lipid vesicles and performs its function for transportation of the lipids in and out to the cells and to the outside. So we can also see from this image that I'm not going to dive into too many details of that one, but the misfolding on APOE4 happens so frequently than the other ones, which puts so much problems on the cellular level. It aggregates and then it starts the oxidative stress for the brain cells, which can cause the death of the cells. The isoforms on APOE4, let's imagine this as an APOE2 protein, this 4-helix bundle, without any of those balls here, which means that it's APOE2. Then after that, if you have the mutation in one of the regions, this one, you get your APOE3, and then the second one is you are having APOE4. The problem is that like the charge accumulation is there. Sometimes there are some structural problems also for this protein affecting its stability and flexibility. So there are some approaches for the therapeutic purposes. One of the strategies is the gene editing, gene therapy, but this is a very highly new area, so we still don't know how to affect the genes and then start producing APOE2 or 3 rather than 4. Another one is the transcriptional pathway editing, which means that you will affect the protein synthesis in the transcription level. And also there are the studies which is showing some evidences that the APOE is binding to DNA and so on. But the most established ways, one of those approaches is that the structural corrector small molecule research. So how it is done is that in these bundles and then the ligand binding site on APOE3, for example, you can see that this ligand binding site is quite flexible and moving around and so on. But for APOE4, the ligand binding site is also interacting strongly with the receptor binding sites as well. So one of the strategies is that putting some sort of a molecule which acts like a lever in between those things and hinder the interaction between those loops, which means that APOE4 is going to gain its function look like APOE3. Another approach is that, for example, this is a small molecule compound seen on the 4-helix bundle, APOE4, found by APOE. So the strategy here is to stabilize the 4-helix bundle and make it less prone to aggregation. Experimental challenges for this protein actually drives us for our intentions for screening small molecule compounds. For example, production, storage, and experimenting on that protein is very hard. It's a delicate one, which is obvious that it's in the brain. So our motivation is to screen compound libraries, firstly to gain the functionality from APOE 4 to APOE 3 look like. And another one is the anti-aggregation propensity, to reduce the aggregation, and then we will have less troubles in the tissue. But before that, for the protein-ligand interactions, I would like to show you how we are actually supposed to see the protein-ligand interactions. So imagine that's your protein, and your ligand is slowly sliding into the active site. There are some changes, complementary interactions forming, and so on. And then after that, we get into our protein-ligand complex. But for screening all of events, we have to use an approach called molecular docking. So this time, you have a protein structure, and you would like your ligand to be bound as like this. So a docking software is trying many conformations of these compounds, and by scoring of these conformations, it gives you some best pose. Okay, this is my binding mode now. So if you have not one, but hundreds, thousands, or millions of compounds, then your docking function or screening pipeline should be able to order by the affinities and give you the best possible compounds, which can be a drug candidate. So for that one, you might have tens or hundreds of millions of compounds. Firstly, obviously, you should go for some property screening, which will give you some information. For example, what are our properties for our tissue, ADME, CNS, BBB permeability, and so on. So then after that, you reduce the number of possible compounds to be screened in a much plausible level. Then after that, we apply geometrical point of view, like the docking functions to screen the compounds geometrically. Are they fitting there? Is there any complementary? And so on. And reduce the number further by applying classical molecular mechanic methods that we now remove the clashes on these compounds, relieve the stress of the binding, and so on. Then after that, finally, we apply semi-empiric quantum mechanical methods which can capture all of these physical effects like electronic interactions, charge transfers, solvation, desolvation, and so on. So then now we can end up with some sort of database of small molecule compounds which can be bearable, can be tested on the laboratories. So the cascading strategy for this screening is just because, for example, in the property screening level, while you are dealing with each compound in millisecond level, for the quantum mechanic level, it can go for hours of evaluation of the same compound. So, which means that we are actually dividing and conquering the data set into more bearable sizes. Our results are that we have screened 2 binding sites identified on this APOE 4. Right now, one was a cavity between 2 tryptophan amino acid residues. Another one is the heparin binding site. And we have screened libraries from FDA Plus World database, which we have 4,500 drug compounds or compounds which are being tested for the purpose of repurposing. So then we can take those drugs and repurpose for some sort of therapy. Another screening done on metabolome databases with sizes of drug-like compounds to identify which parts of the proteins are doing what kind of interactions with the molecules which are available in the brain. So we want to correct the function of the protein, but without disturbing its function. So that's why we are also trying to create some sort of information about its function as well. Then we are also going for multi-million compound databases like zinc or enamine to be able to find novel scaffolds which can specifically bind on that protein without disturbing any other systems. So, so far we have purchased 25 compounds from our screenings, and 10 of them showed anti-aggregation functions on the protein. And my colleagues are also evaluating the protein function in nanodiscs, so-called lipid vesicle kind of systems, to also confirm that APOE4 is not losing its ability to form lipid vesicles. So then we will have around 35 compounds to be purchased later. But the take-home message for us is that so far we can able to reach to screen around 15 million compounds. These are individual binding sites, so 15 million every time. Another thing is that aggregation doesn't mean that only binding equals activity. We actually have more compounds identified as binders, around 20, but 10 of them showed this aggregation propensity reductions. So we can able to identify binders really with a high rate. Another thing is that the computational challenges, so far the APOE doesn't have a very distinct cavity. So what we have to do is to push the boundaries of the methods to be able to identify even the weakest possible interactions. That means that you have to go for very high level calculations, which means they are expensive and they are time consuming. As an outlook, now we are also aiming at the screenings on the mutation side, as well as searching for any other possible binding sites or cryptic pockets. So far in the future, maybe we can also figure out some null binders. Another aim is to go for larger databases, which means maybe billions in size, better property screening, a better pipeline for evaluating more compounds in batches. One of the aims is to evaluate the lipid maps structure database, which is important for us because the dry weight of the brain is around 70% of fat, and that's the most important thing. Is the lipid metabolism is important for our brain. But for example, one of those smallest compounds from screenings in the literature is something like this, with a very less number of degrees of freedom in conformational point of view. But when you look at some of the lipids, it's like a monster for us. To do that, it's just not quite possible to use our pipelines in this physics-based heavy machinery to identify binders or understand the functions of these compounds. But we are also training ML models to speed up the progress on the screening. So far, our preliminary studies showed us some 250-fold of increase in speed. I'm sure that we can even push it further. And this is without loss in accuracy. It actually acts like how SQM or quantum mechanics do, with a very high generalization. We are also planning for a fragment-based drug design, which means that our small molecules are going to be screened and grown inside our detected cavities. And this is all about how we can apply these methods for quantum computing purposes as well. So we are trying to crack the quantum computing work for us for screening of these compounds. And in our laboratory, we are not only specialized with those screenings, it's actually specialized with protein engineering as well. We have many of these nice protein engineering tools for research, and they are freely accessible for everybody in the academy. And it wouldn't be also possible without my colleagues, experimental colleagues from Loschmidt laboratories and colleagues from collaborations here from CIIRC, and without our institutions. And I really would like to thank you for your attention. And I'm really full of ideas between those 2 days, because I met with many people, how to work with, for example, on multiple targets, because there are multiple reasons for the Alzheimer's disease, or maybe not a multiple targets, but screen the possible compounds with the virtual human twin by all of these control points in the biochemical pathway of humans. So it's really interesting here to give you a talk, and I really appreciate your attention. Thank you.
Thank you so much. Thank you. I'm not going to pretend I understood most of that, but it was so interesting. Think about it. It took us 2,000 years from Democritus to Avogadro to get from atoms to molecules, and it took us less than 200 years to get from the invention of molecules to this wonderful methodology. So congratulations, it's very impressive, and thank you for all the work that you do. Now is the time for some questions from the audience, and let's have a look if we've got any on the screen, please. None. There's a sad head shake from the back. That is very disappointing. Please, I'll give you a microphone and you can ask in person. Oh no, the ladies bring the microphone. Wonderful, please.
Thank you, great talk. You closed with a lot of sort of next steps and future looking from a methodology perspective. But I think partway through, you described the results of what you've done already with your existing methodology, and you ended up with, I think, around 20 compounds that you, I think, were saying were good candidates of actual molecules. So my question is, what are the next steps for those candidate molecules now? Are you looking to partner with other parties who can do some experimental work with them?
There are still ongoing experiments on those compounds. So since in Alzheimer's there are lots of aspects, also for these proteins as well. So we developed our pipeline for screening. We are still developing further for making more efficient and more aware for any kind of physical realities. But at the same time, the experimental part of our laboratory is developing their own pipelines, because APOE is something really hard to handle. Because you have to make some experiment on a protein, but this is not something that you extracted from a plant. Its environment is quite unique, so you have to put there as much reality as possible. For example, the lipid vesicles. You have to make the lipid compositions in such a way that they exist in brain, and so on. So there are still ongoing studies on these compounds. Not in the phase that we will find some partners or we will go for.
Yeah, I was thinking even in vivo. And part of the reason I'm asking is some former colleagues of mine, when I was in pharma, I know were very interested in APOE as a target, as a drug target. The company I was with, we never actually had any programs that got off the ground for that, but I know there are some very sort of senior placed people in institutions in the US that are, have a historic interest in APOE as a target, so that's sprung to mind.
Yeah, okay, thank you.
Thank you, and you've actually foreshadowed a fantastic thing for the panel discussion which will be happening, the quadruple helix, where there will be people both from the private side of things and the public side of things and academia. So I think that this kind of thing that you mentioned, Big Pharma, for example, has seen the application of this research is going to be definitely part of that discussion. So thank you for inadvertently foreshadowing that. We have a question from Slido. Thank you. Is the active site of a protein rigid or flexible?
It is flexible. If we keep the active site of the protein rigid, I'm sure we can't find anything, because we need to have as much features as possible from the protein site to be captured. There's no active site actually. So it's just binding site. And even we call it further like landing sites, because it's so shallow that you just can't dive into some place. It's just you land on it. like landing this the lander to the Rosetta Stone comet. So it's something like that. There's no gravity, there's no strong interactions, and so on.
Thank you so much. Ladies and gentlemen, you can definitely grab this fine young gentleman gently during the networking afterwards. Unfortunately, for now, we will have to move along. But please, one more round of applause for Saltuk. Congratulations on a wonderful talk. Thank you. I was actually talking to a cognitive anthropologist friend of mine, and he said that he's got a massive problem with clever people being diagnosed with Alzheimer's because they don't want to admit that they didn't get the test results well enough, so they kind of memorize the answers in advance, and therefore it's actually a lot more difficult to diagnose dementia with high IQ people because of the ego. So it's quite interesting, the qualitative side of that. However, we heard about the first research program track. Now it's the time for the second one, and the person representing that will be Michal Seják from the Department of Cognitive Systems and Neurosciences from this fantastic institution, the Center of Informatics, Robotics and Cybernetics, the Czech Technical University. Welcome, please, round of applause.
Thank you very much. Thanks for the introduction. It's an honor to be here. My name is Michal, and I would like to present OpenSpindleNet, which is a sleep spindle detection method in skull and intracranial EEG. So first we're going to talk about what EEG even is. Why do we care about sleep spindles? And then I'm going to enumerate the data sets we used, show you the architecture I've went for in this research, and then I'll show you the results, where we got, and what does that leave us with. Right, so what even is EEG? So EEG, I'm sure some of you have seen this signal. It's basically a capture of the brain activity, right? So it's a recording of the electrophysiological activity in the brain. And polysomnography, PSG, which is possibly a new term for some of you, is when you add sleep study-related stuff to it. So when you measure breathing as well, or eye movement. PSG relates to how the whole body functions during sleep. And in sleep, in a specific stage of sleep, these sleep spindles, which is a specific artifact, occurs on the EEG. And sleep doctors know that if you have sufficient amount of these sleep spindles during your sleep, then your brain is healthy. And if you don't, then there is a problem that they have to solve. I am not a sleep doctor, so I'm not going to be able to tell you what these problems are, but I try to detect these automatically. Because if you have hours and hours of such signals, then manually going through them and trying to find if there are spindles or not is very labor intensive. And it's also subjective. Many doctors do not agree whether a part of the signal is a sleep spindle or not. So for large-scale research, you need to have automated methods which are able to detect these spindles. And on the right-hand side, you can see an example of this spindle. It's an oscillating pattern. And these patterns are usually 11 to 16 hertz and about a second long. Another problem is if you have the EEG coming from a scalp, which means the electrodes are directly on your head, then the spindles look different than when these electrodes are implanted directly inside of your skull. The scalp EEGs are, the scalp spindles are subtle. They usually have a low signal-to-noise ratio, and in eye EEG, the spindles are a high amplitude and high SNR. And they also have a variable morphology. You can see that all of these spindles look very, very different. So we want to create a robust tool for automatically detecting these spindles, and also want to create an architecture that is able to detect EEG spindles as well as eye EEG spindles, right? And we want to make this tool open source so we can accelerate research not only in our lab, but also beyond. The data sets we used for this research were twofold. We had an internal IEEG data set, which we collected directly in our lab. And then we also used a public SCALP EEG data set because we want to be able to show that other methods are, how well do they compare with our new method. You can see a short overview of how large these data sets were. You can see that in our IEEG data set on the left-hand side, we only have 4 patients. So we were not able to create a good patient-level split, right? So we are not sufficiently confident that our method will generalize well to new patients, but this is a good first step, let's say. And also to ensure high-quality ground truth in this new data set that we built, we implemented an iterative labeling approach, right, where we created a first iteration of our model, then we used it to label some data, and then we used an external reviewer to review these potential labels, and this resulted in us basically quadrupling the amount of labels in the data. It was just a single iteration, but still we created many more new labels. So this is the data we worked with. The architecture we used a dual input and a dual output. The 2 inputs that went into the detector was the raw signal, which we normalized, and then we built a scalegram out of the signal. So the normalized signal basically shows you the shape, and the scalegram shows you the spectral power of the signal, right? So we wanted to give good features or interesting features for the network. For the backbone, we used CDIL, which is a good convolutional 1D network. And for the outputs, we had a dual head, which means we basically had the network output 2 kinds of outputs, right? We had detections, which was like a YOLO head detection model, detection head, which tells you if there are spindles and where are they, how large are they. And then we had a unit-like segmentation head on the output, which basically tells you for every single sample if it is a part of the spindle or not. Right, so to repeat that, sorry. The detection head, you can see the output on the top, right? You have these intervals, which you can basically see if there are there or not. And for the segmentation, you have a prediction for every single time step. And it can or cannot possibly match with the label that you expect. Right? So the detection head basically tells you where do spindles start, where do they end, and the segmentation head tells you which samples belong to a spindle. And then you can ask Michal, why do you do both of these tasks at once, right? It doesn't make sense. Why do we need both of these outputs? The trick is we basically apply a synergy through multitask learning. We only care about detections. We only care about detecting those spindles, but training both the detection and the segmentation head gives you a better representation on the backbone and in the rest of the network. So the segmentation head basically works as a regularizer. Sorry. How do we evaluate that? How do we say if our method is good or bad, right? In the state of the art and in prior art, there are basically 2 ways of evaluating this model, right? So if you have some labels in green at the top and some predictions, you have 2 ways of saying whether you are doing a good job or not. In the detection way, you calculate an intersection over union between the label and the prediction, and if they are high enough, then you say, okay, this is a true positive, this is a hit, and if not, then you have a false positive or a false negative possibly, right? So this is an event counting method. And then you can also have a sample-based counting method, where you look at every single time step and judge it, whether it's part of a prediction or a label, or both. And we prioritize the sample-based method because it encourages the model to learn more precise boundaries, right? And it's also more prominent in prior art. So we went for the second way. And then we did an ablation study on the dual output, right? Because you could say, Michael, I don't trust that this segmentation head does anything for you, right? So that's a fair point. We tried the detection-only method. And then for the bottleneck, which is a part of, at the end of the architecture, we could either share it between the heads, so like in a Siamese setup, or you could train it separately. And we tried all of these different methods and trained the hyperparameters for each. And then using statistical testing, we found that the separate bottleneck variant works the best. The separate bottleneck was slightly less sensitive in raw detection F-measure, but it was superior in segmentation F-measure and also in AP in detections. So it was actually better in both the tasks. The results on the IEEG were pretty solid. We tried to compare our model with the public available methods that we could gather. And we turned out that we actually achieved the best scores. So we found a new state-of-the-art on the new data set that we just created, which is not that impressive, right? But we can see that the model works well for IEEG. And this is how the predictions can look like, right? So if you take a look at the bottom, you can see examples of a true positive detection and then a false positive and a false negative, where the labeler found a spindle, but the model didn't see it because it was not prominent enough. And on the other hand, the signal had some characteristics of a spindle, but it was not actually a spindle, so that's where the model made a mistake. And for the EEG, for the DREAMS dataset, for the other dataset, where there is a lot of prior art, we scored, again, at the top. We had the best detection F-measure. We did not have the best segmentation F-measure. Some methods actually were slightly better, but these methods had manually tuned parameters on the test set. So there is a very high probability, or a higher probability, that they will generalize poorly. So we did not become too sad about that. And so we found that the architecture is competitive and performs on par with top-tier methods. And again, we can see an example of some predictions of a bunch of true positives and then a false positive, where the model was very confident that it sees a spindle in the middle, but the labeler did not say it is a spindle. The question is whether the labeler was correct or not, because in such data sets, right, you do not have perfect ground truths. So even the expert that checks the labels can sometimes be wrong. And finally, we also measured the characteristics of these spindles, right? Because if you have good F-measure, it doesn't mean too much if the events do not look like spindles, right? So we measured the amplitude, the frequency, and the duration of the expert labels and our own detections. And we saw that the amplitude and frequency matches, roughly, but the duration of our model is slightly too large and a bit too rigid. And this is an architectural drawback that we are actively working on right now. So we found that the characteristics closely match, but the duration is a bit too rigid, off by roughly 10%. So in conclusion, we developed a novel deep learning architecture that detects spindles in IEEG, and we also showed that the architecture generalizes pretty well to EEG as well. We also showed that the dual head design was statistically superior, so it made sense to do this. and the tool is open source, free to use. It can be installed as a Python package, which bundles the ONNX exported model and all the code that you need to run this thing. Right, so that's it from me. Thank you very much for your attention. Thanks to my colleagues that made this work possible, and thank you for your attention.
Thank you so much. My friend actually works in diagnosing epilepsy for children, and yeah, I think I'll send him your way because there's a lot of fantastic things that you uncovered. Ladies and gentlemen, any questions from the audience? My obligatory question. No? Yes, all right. Some live ones, good. If you're shy, use Slido, don't worry. But if you're not shy, you can ask live. Thank you.
Thank you for your beautiful presentation. I have one question. Is there a link, a significant link between dementia and anomaly in the EEG, or the IEEG, or the presence of spindles?
That's a great question, and we actually, in our study, we did not study the impact of illnesses on the quality of this data. Actually, all of the IEEG data that we had, our own data set, were composed of epilepsy patients. And those had this specific procedure where we implanted these electrodes done, right? So these are very specific patients. And the tool can now be used for, running it on data that that there are dementia patients possibly, and automatically, find these spindles and then test these characteristics instead of having a doctor go through them and manually find the spindles, then compute the characteristics.
Thank you very much.
Thanks very much.
Thank you. Please.
Nice presentation, and I liked your comment you made at the end that the human ratings might be, there's something to develop against, but it's then worth having that feedback loop because especially there's so much data to go through, they may have missed and they may, so something like a self-overread or an overread is an important step because otherwise you may be optimizing too much on something that inherently has some variability. But my main question was more of a devil's advocate question, and as you were presenting this very impressive set of work, as I just eyeballing the EEG traces, I was wondering whether your methodology was a bit of a sledgehammer to crack a nut, in the sense that just looking at the traces, it would seem to me that you could do a pretty good job based on a sort of old-fashioned rules-based approach, based on frequency decomposition, different time scales, sliding window, or what have you, and then some decision rules based on that. Is that naive, or have you compared your performance with sort of a noddy non-machine learning performance, and how much better is your approach than sort of old-fashioned approaches?
That's a great question. So primarily why we even started working on this, because we needed a spindle detector that worked in IEEG, and we tried all the available methods and they failed. We saw on one of those slides the comparison between YASA and Sumo A7. Those are like the basic or the naive, simple CV-based methods that do not use machine learning, and they had a much worse performance than this method that was based on deep learning. And actually, it could be kind of scary, right? Using a sledgehammer on this task, but we try to build the model in such a way that it's very small. It actually doesn't even have a megabyte. It's in size of hundreds of kilobytes. So it's a very small model that you can run on CPUs quite simply, and you have a very fast inference time.
Yeah, that's a good answer. I think when you present this going forward, I think make it clear that those other models are not other machine learning models, because it wasn't obvious to me.
Yeah, sorry, yeah, that's a fair point.
Thank you. Yes, one more question, sorry, to your left, thank you.
Do you use some fixed sliding window as input of CWT, or you do use some K-means to set the width of window?
No, we used a fixed sliding window. It was considered as a hyperparameter to tune, but we had so many other hyperparameters like sizes of the network at various stages, etc. So we decided to use just one that fit the expected spindle frequency range, right? The spindles are expected to be in between 11 to 16 hertz. So we used a slightly larger, with a buffer window above this, and we used Shan 613 basic wavelet filter, and we got this.
So you have to know sampling frequency of your data input.
Yeah, we have the model works on a fixed sampling frequency that it's known beforehand. So if you have a different sampling frequency, then you just resample and then you use our model.
Thank you. Thank you very much. An excellent answer. You should be in talk shows or something. Very good answers as well. Right, any more questions? No? All right, I'd also like to liven this up a bit. Where do we have all the students from the Ukraine? Hello, are you enjoying yourselves? Yes, you have to nod enthusiastically. Yes, fantastic. We're very happy to have you here, and I hope that you're enjoying today. And also, thank you so much to Clara and everyone for making this happen. So we're a very lively audience of all ages, and please give a very youthful, happy, loud round of applause to Michal for his wonderful work. Thank you.
Thanks very much.
Thank you so much. Thank you. There was a very important keyword mentioned of the sledgehammer, and I think it's interesting that in a lot of discussions, AI and machine learning is very much being considered as a very kind of heavy tool because the computing power that is required to make it happen is very, very large. But that is one of the things that we want to present to you today. You had the opportunity to see the Research Program 1 and Research Program 2. But how about all the metal that all this runs on so that we don't run into an AI winter and we can actually make all this computation happen? To talk about this is on the horizontal topic is none other than Luigi Iapichino, who is the Group Lead User Enablement and Applications Quantum Computing and Technologies Department. Please welcome to the stage. And a round of applause again. Thank you. The floor is yours.
Thank you. Yeah, especially today, we have seen lots of research on the topics which concerns the CLARA project. So what we want to do now is to have a closer look also at the infrastructure that we can have available for them. So in terms of, speaking of making this a little bit more entertaining, I was thinking, well, just having a list of things that we can make available to researchers, this would have resulted maybe a bit dry. So I was thinking to follow a slightly different approach. Namely, I will show how the evolution has been done between the high-performance computing as it was, I would say, about a decade ago, so a really monolithic one, towards something more heterogeneous, which in fact is what we make available for this project. So from a monolithic approach to high-performance computing, we will move to a modular one, where AI and machine learning is part of this approach. Quantum computing is also part as a new class of accelerators in high-performance computing, and then we will just give a few hints about what the CLARA researchers have available for them. Yeah, let's start with a very concrete example. So this is SuperMUC-NG, the flagship system at the Leibniz Supercomputing Center. It's not the newest system. Is it working? Well, doesn't matter. It's not the newest system that you can see. This has been inaugurated in 2019, so we have already the successor system on the pipeline for 2027. Well, with a high-performance computer like this, you can do many different things. So let's say, for simplicity, whatever you cannot do with pen and paper, you can do on these systems. You can have applications on many different areas. I'm an astrophysicist, so astrophysics is one of the main ones. You have computational fluid dynamics, molecular dynamics, relevant for here, right? And many other fields. And one of the key of using such systems on such a broad range of topics is that this system in particular has a very good usability. SuperMUC-NG has been conceived in a time, so say, bit more than 5 years ago, when it was still possible to have a system without accelerators like the GPUs. Such systems, so such non-accelerated systems, were extremely good in serving a broad range of user communities. Nowadays, this is not possible anymore to do a system like this for many different reasons. The main one is that both in terms of performance and in terms of energy savings, so if you want to keep up with offering more computing power to your users, you need a system which, on the one side, needs some energy efficient modules like the graphical processing units, the GPUs. On the other side, we go from a monolithic model where one system can serve different needs, different communities, to a model where the system is going to be so-called modular. So there are different parts of the system fitting different needs, and at the end of the day, also different communities. So we are talking about GPUs, for example. GPUs are great for machine learning AI applications, for example. And with the system, with different modules, with different components, one can cover a broad range of applications. So we see in HPC two different worlds that basically went together. We go from the classical HPC, where one had the other modeling and simulations, basically starting from the equations, making them into algorithms and analyzing the outcomes of simulations, to the world of AI machine learning, where the discovery is driven directly by the data, and this can be seen also, big data is part of this. There is a convergence of these two worlds, given, for example, by the usage of GPUs, among many other things. Let me just provide a few concrete examples. And here, again, I'm not an expert of these scientific communities, so I will give a very biased list of works which I think can provide good examples of the trends that I want to highlight. The first one, as I said, we start from a case with a non-accelerated system. This is something that has been done very well. So the Gordon Bell Prize, the Supercomputing Conference in 2016, the Gordon Bell Prize, yeah, if you are not familiar with that environment, it's like the Nobel Prize for high-performance computing, right? It's awarded every year at the biggest HPC conference in the US, the Supercomputing Conference. This went into a study of molecular dynamics done on a completely CPU-only system in 2016. Since that time, many things have changed. GPUs, machine learning technique get important. Very recent study in this case, done by a Chinese group, where techniques initially developed for machine learning are applied to simulations of quantum chemistry. So this is where the two things, to some extent, tend to converge, right? Now, if we talk about HPC, we talk about AI, we want to enter into this paradigm also quantum computing. And here, of course, for reasons of time, I cannot go deep, very deep in quantum computing. We can have a discussion afterwards. I'm here until the end of the event. Let me just mention a few key important things. So quantum computing, from our viewpoint, from a perspective of data center, is not important as a standalone tool. This is just a new class of accelerators for specific workloads in HPC. This means that for just a few but important class of problems within a broader HPC workflow, it makes sense to have such computations offloaded to a quantum computer and the results back then to the HPC workflow. This is the way we want to work, integrating HPC and quantum computing from the hardware viewpoint, from the programming viewpoint, from the software stack viewpoint. And from the perspective of a quantum computer, the integration is also important. And here I'm really diving for a moment deeper in quantum. So the system that you have now in quantum computer are small. Well, they say intermediate scale, but they are small. Noisy, such that basically whatever you can run there is very short, is shallow. So you cannot have very long circuits because the system is not able to handle them. So you need short circuits. In order to have short circuits, what you do basically is to have a kind of iterative process, as it is shown here in this plot, where you submit to the quantum hardware parametric circuits, you get some result, you pass this result to a classical optimizer, which updates the parameters in the circuit, and you go back and forth until you reach some kind of convergence. These so-called variational algorithms are the ways that we have now to work on quantum hardware. This is useful because one can already do useful work on the systems as they are today. We can have them already integrated into classical computing, HPC, so we don't want them have integrated on my laptop. We want to have integrated into an HPC system because we expect the size and the quality of the quantum hardware to improve. At some point, we will move to the so-called fault-tolerant quantum computing, where there will be protocols to reduce even more the noise on quantum hardware. In that case, those protocols will have to run on HPC systems themselves. So for this reason, this integration, also from a quantum perspective, is very important to us. And here, again, let's move to a few examples of this. And again, the choice is biased. The first study is a proof of concept of integrated HPC and quantum in a team which is also involving our collaborators from the University College London and my team at LRZ. This interplay is interesting not only as a proof of concept, but also at scale. So there is a recent paper done on the Fugaku supercomputing in Japan, together with IBM, where it was possible to have in the same workflow the full Fugaku system together with a quantum computer, so not just the computing power of a laptop, namely one of the biggest HPC systems of the world. And in this evolution, also the data-driven discovery enters the stage. We have here, for example, a work on a dataset for protein-protein interaction from a team involving also CLARA partners. So that's a full story that is going to converge here. HPC, quantum techniques from machine learning and data are all here. What I'm going to claim here is that for all these different needs, for all these different tools, we have computing resources available in CLARA that can address all these different and complementary needs. So we have high-end HPC systems, we have quantum hardware, and we have the Lexis platform, as I'm going to show in a moment, which is basically the tool for accessing, orchestrating workflows, managing the data. In terms of quantum hardware, at the Leibniz Supercomputing Center, we have two systems that we make available for project partners. One of them is QExa. So this is a superconducting system with 20 qubits. It's the first, to our knowledge, quantum hardware co-located in our HPC infrastructure in a data center environment. This has been run for test users in 2024. Since September 25, we are in the regular user operation, meaning that all users can submit a project proposal and access the system. The access has been provided to CLARA partners. We have also, on our HPC resources, a noise model deployed so that the users can test on HPC resources in advance to access in the system if what they want to do makes sense for the features of the system. A successor of QExa is EuroQExa. This is a co-funding initiative at European level, federal German level, and the state of Bavaria. This comes into larger systems than QExa. The first one, the 54-qubit system, is nearly close to be open to test users. So the system is already installed. We are waiting for the final green lights to enter these in test user operation. This is available for European users, so CLARA researchers are entitled to use that. We, as a matter of fact, we received already a use case from CLARA researchers to enter the first group of test users of EuroQExa, and I think this is something that would be important also for us to have these use base so aligned with our common projects. Then, as I said, the Lexis platform. So this is something that also in the framework of this project is made available to this community. So this means basically accessing easily. So I will not go much into the details here, but just what does it mean for our community? So getting access to HPC resources, to quantum computing, to AI, within a framework, a platform that can allocate all these needs, orchestrating the workflows, distributing the data in a fair way, and having basically a web portal where one can access all this system and track the execution in this way. And basically the final goal, the final vision that we can have with the Lexis platform is that all the HPC resources, the data resources, the quantum computing resources, through all our partners will be basically accessible through this platform to our user community. And with that, I think I can conclude here and open for questions. Thanks.
Wonderful and extremely enriching and wow, definitely a great end to the session. Ladies and gentlemen, any questions? Oh, you're brushing your hair. I thought you were putting your hand up. Ah, that was deceptive. Anything in Slido? Nope. It's always the sad head shake from them. Come on, make them happy. Ask a question. No? Yes? Ah, yes, there is a question. Fantastic. Thank you.
Thank you, Sarah. So we were speaking about collaboration, so now, you have access to the computational power. So speak to Luigi and compute, that's it.
Yes.
Sure.
Thank you.
That's, please come to me. Well, I know that my name is not the easiest, but you can write an email to quantum@lrz.de, which is much easier. And yeah, you can reach out to me and we can see how we can support you.
Thank you very much. Ladies and gentlemen, another round of applause for Luigi. Thank you.